สวัสดีครับอาจารย์
สวัสดีครับ So we miss only one of us, right? I forgot his name, Dr.
Shun.
Dr. Shun, right? So the students in Thailand are all ready. There are 4, right? Shall we start?
Yes อาจารย์ you can start.
Okay. Last time, I hope I forgot to use the animation for the slides, so the sequence is a little bit like going rough a little bit. But now I hope I will start begin this lecture a little bit faster, and there are not too much content, and I will try to talk only the highlight. Last time we have overview that the immunity is the system to protect us, not just from the outside, but from anything harmful inside our body, including the cancer cells or the dead tissue in our body as well. For the outside, it's like the foreign body, and as well as the microorganisms. And our innate immune system, we are divided into broadly 2 types: innate immunity and the adaptive immunity. And there are some link between the innate immunity to transfer into the more adaptive, more specific, and more coronal expansion with the memory to the adaptive. That link requires the antigen presenting cells and some cytokine signalings. I talk about the several cytokines, and you should know the one. I mean, there are several cytokines that you should know, like the cysteine-rich ligands, as well as the chemokines, and other and interleukins. And you should learn some importance of other cascade proteins called complement as well. That is a part of the innate immunity, and I will talk again in this lecture. The rest of the class, the subject, I think they will go deeper into the detail about the roles of the adaptive immunity. But this lecture, let's focus to what is in the beginning. The innate immunity begins at birth, and I already described about the natural protective barrier. That's the physical chemical barrier. That is also a part of the innate immunity. And other several cells and several chemicals are responsible for the innate immunity as well. These are just the first line of our defense. So it gives the initial response, and it prevents, controls, and eliminates. Mostly, the function of the innate immunity just mostly protect us from the foreign bodies and infection. We'll talk about more about the roles of the innate immunity in the infection. It also eliminates damaged cells and initiate tissue repair. And there are some link from transfer from signaling from the innate immunity to the adaptive immune responses. The innate immunity, there are 2 main functions of the innate immunity. There are two legs. One is initiate inflammation and signaling for the adaptive immune response, usually for the extracellular signaling. And the other one, the other leg, the innate immunity will give us the initial response to the virus called the antiviral defense. An outline of this talk, we learned a little bit last lecture that you already know that the adaptive immunity, they can adapt and very quite versatile. In innate immunity there is a very limited kind of response, but during the evolution, our innate immune system also recognize the most common pattern, like response to the virus, response to the bacteria, special kind of bacteria, recognize special foreign molecules that we don't normally use, like mannose. We know that we have a lot of sugar, but our human body, we don't use much of the mannose. So if other organism has like mannose sugar, we can recognize it as a foreign. So it means that innate immunity also has some variety of response, but they are response as a pattern. So we have patterns like or the recognition of the molecules, and we will learn some of them, like 10 pattern recognition, and there are some abbreviation, ตัวย่อ of the pattern recognitions of these molecules. We don't talk much about the barriers, we already know, but you should know that there are several cells. In the beginning, I talk about the tissue responsive cells and phagocytes like neutrophils and macrophage. But there are some other cells that you should know in this lecture as well that are responsible for the innate immunity. And we will explore a little bit about the effector molecules involved in the innate immunity and some of the molecules that send signals for the further to talk with the adaptive immunities. As I told you that the innate immunity responsible for most of the inflammatory process and the antiviral defenses. Okay, talk about that. And somehow the innate immunity cannot go on and on forever. There should have some stop signal that you should know. Okay, let's recall the difference between the innate immunity and adaptive immunity. So innate immunity actually also has some specificity and diversity as well, but the specificity and diversity is limited. It doesn't mean that it has no specificity or diversity at all. There are, but the amount of the response is limited. And because we have to fight with bacteria, we have to fight with fungus for our generations. So usually they can recognize the patterns of our foreigners, like bacteria or fungus as well through evolution. So just to compare with adaptive immunity, there's so diverse, so specific, and can proliferate a lot, but the innate immunity is limited. So the picture on the left-hand side, you see some patterns from bacteria or some patterns from the virus or fungus can trigger. We have the responsive molecules or the receptor molecules for the common bacterial membrane. The ones of the best known, we call it toll-like receptor. And we have our body don't produce mannose, but we have to have like mannose receptor to recognize that the invaders are foreign to us. That is, for example. Okay, these tables show that there are a lot of the patterns that develop that our evolution of human or our evolution of animal can recognize the foreign thing that is harmful to us. If it is come from outside, we recognize them as patterns. So as pathogens, and these patterns of recognition of the invaders commonly, for example, our cells are intact, so we don't expose RNA or DNA easily. So if there are RNA or DNA that come from outside, probably is come from the virus. Okay, so we have a receptor for this nucleic acid, like recognize the single strand RNA or recognize the double strand RNA, or recognize certain base repeats like cytosine and guanine repeats. If we recognize this bacteria, that means that there are some invaders against us. Also the proteins, the tail of the microorganism we don't have. So if there is the tail of the microorganisms come to our body, we can have a receptor that fights against it. Okay, and we don't have lipoproteins or LPS, lipopolysaccharide. We don't have that kind of thing. So if it comes to us, it means that it's foreign. These are pathogens, mannose or glucans. Our body don't have it. And due to the evolution, we have receptors to act against this kind of thing. If it is come from outside, we call it pathogen-associated molecular patterns, or PAMs. And if it's like, normally we don't produce this kind of proteins, but when we will produce this kind of protein when we damage, self-damage, anything wrong happen, like we have stress, the cells have some stress, the heat shock protein might be trigger the inflammation, or the urate crystal in gout arthritis. We can produce it, but we don't normally produce it. And if it is too much, it can cause the disease. We must get rid of it. We have to recognize it. Proteoglycan peptides, formulate peptides, and histone usually is in the nucleus. But if it comes out, it means that our cells can go wrong. Our cells may get some damage, and when we need to get rid of these damaged particles. So we call these things are called as DAMs, or damage associated molecular patterns. So you know that the PAMs and DAMs, when it gets inside something wrong, it's not normal to our body. So how can we recognize? We have to have the receptor to recognize this foreign or possible harmful molecules to us. And the PAMs has some receptors, the DAMs has some receptors, and we don't group, we can group according to the pathogen or the damaged molecules inside, but there are some other ways of grouping of the pattern recognition receptor, or PRR. One of the best known of the pattern PRR to recognize PAMs and DAMs, we call toll-like receptor. It's like ด่านทางด่วนอ่ะด่านเก็บภาษีเขาเรียกโทรนะเก็บตังค์ It's like if you drive a car to the expressway, there are some toll booths to collect your money. So that is the receptor stand for toll-like receptor. And toll-like receptor or TLRs, there are two types. One is expressed outside. If you see on the picture in the representing the cells, TLR, some of them expressed just as the transmembrane proteins. There's and signal to the cytosol. And there are some toll-like receptor that recognize something that we already ingested in, is the endosomal toll-like receptor. Okay, there are numbers like 20 of them, 20s or 20s of them of the toll-like receptors. But broadly, we categorize into two types: toll-like receptor on the cell surface and the toll-like receptors in the endosome. And we also have other different pattern recognition receptors which are floating in the cytoplasm or the cytosolic receptor, just to pass it, just like NOT-like receptor, RIG-like receptor, and so on, to recognize different PAMs and different DAMs in our body. We also have the mannose recognition receptor. We call it lectins or dectins or scavenger receptor when we got some damage, or N-formylmethylusin receptor. That's the minor things. What I would like to emphasize, like two things. One is that in order to recognize PAMs and in order to recognize DAMs, you need pattern recognition receptors. The only receptor you should know is the roles of the toll-like receptors. Okay. So we talk about a little bit more about the toll-like receptors. It has the when foreigners or damaged molecules trigger the toll-like receptor, the toll-like receptor will stimulate a lot of enzymes inside our cell. But they are broadly categorized into two downstream effect function. One is if, one moment, there's a phone coming. Okay, sorry. One, as I told you that at a cellular level, the innate immune response will either respond to the causing the inflammation or cause the antiviral activities before it's sending to the adaptive immune response. If it's the effects of the toll-like receptor signaling to cause the inflammatory response, it uses special transcription factors called NF-kappa-B, nuclear factor kappa-B, or the AP1 and AP1 proteins. But if the toll-like receptor wants to have the effector to the antiviral defense, usually it acts via the action of interferons. So it needs special transcription factors called IRFs, which stand for the interferon regulatory factors. The most common ones are IRF3 and IRF7. I am bad at remembering number, but usually just want to tell you that the pattern, the the best known pattern recognition receptor are toll-like receptor, and toll-like receptor has two effects. One is inflammation, and one is antivirus. The ones that goes to the inflammation, it needs new transcription factor called nuclear factor kappa B, NF kappa B. And if it's mainly act for the antivirals and new interferon function, it acts via the IRF transcription factor. Okay, we talk about the innate natural barrier a little bit, that our epithelium and the skin can has the innate immune response by having the physical barriers, but not so. It's not just the epithelium act as the physical barriers. These most of the epithelium also produce some proteins that to protect us from harmful invaders, harmful bacteria. Sometimes it can produce proteins harm to the invaders as well. Those examples are defensins, catalycidins. Just to let you know, you don't have to remember that it's not just the physical barriers. These can produce some proteins toxic to the microorganism as well. And from the first lecture, we know that most of the lymphocytes are adaptive immune response, but there are some exceptions. There are some special T lymphocytes, there are some special B lymphocytes that has limited response, and those usually limited to the innate function. For example, some T lymphocytes only resides in the epithelium. We call it intra-epithelium T lymphocytes. It is T lymphocytes, but it is not adaptive. It has the limited T cell receptor specificity. So this is more to the innate immunity. But how do we know that it is T cell? Because it is CD3 positive. If it is CD3 positive, it is T cell. Okay. And as you already know that phagocytes, if the tissue macrophage, if it is already there, it eats all the invaders. And if it is circulating like monocytes, it can travel and doing the complex differentiation. As well as neutrophils, it can do the phagocytic function. In the innate immune response, sorry. There are some other cells in the innate immune response. One of them you already know, dendritic cells. I talked a lot in the introduction part. I don't want to talk much now. What I'm going to, the dendritic cells are important antigen presenting cells, as I told you. The next type of cell, they are good at killing invaders, especially virus. We call them natural killer cells or NK cells. The NK cells, they do two functions against the virus. One is they signal in the figure A. They signal they recognize the injured cells or the viral infected cells, and then cause them to get the apoptosis. It's like the CD8 T cell, but this is the innate immune cell. And also, if the NK cell cannot kill directly to the viral infected particles, they can receive the stimulation of the interleukins and produce the interferon gamma. The interferon gamma then signaling to the killing of the phagocytous microbes later on. So either they have direct signal to the killing of the intercell, or they produce the interferon gamma. This is the function of the innate immune cells called the NK cells. Okay. We know some of those cells. Oh, there are some other cells. Just briefly, that there are a lot of immune cells that in the innate immune response, other than the phagocytic cells and dendritic cells. Just to let you know that innate immune cells also has the innate lymphoid cells, the cells that look like lymphoid but is not fully adaptive cells. It is similar. Some of the innate lymphoid cells, they are similar to T helper cells or the CD4 plus cells. It is look like, somehow it's look like the T helper 1, sometimes it look like T helper 2 or T helper 17, but they cannot fully call T cell because they don't have T cell receptors. So it look like lymphocytes, but it is not. So we call them innate lymphoid cells, acting against invaders like lymphocytes, but the receptor is not diverse enough. There are some T and B lymphocytes with limited antigen receptor specificity, like iNKT or mucosal-associated T. Just quickly, just to let you know and familiarize with it, but you don't have to remember. Just to know that innate immunity also has the cells that look like lymphocytes, or they have the lymphocytes but limited response. Mast cells is also part of the innate immunity and cause a lot of inflammation and hypersensitivity reaction, and release a lot of pro-inflammatory cytokines and histamines and lipid mediators. Okay. Apart from the cytokines, the innate immunity and the innate immunity also has special proteins that you might want to learn, like the collectins and ficolins. You can go and read about it in the text later on. There are some limited clone of the and less specific antibodies produced within the innate immunity, like natural antibodies. Natural antibodies means that the antibody is produced without having, you don't need to pre-expose to the antigen, but these lymphocytes just produce the already produced in our body, and most are IgM. You know forget it. You will learn about the IgG, IgM later on. I won't talk to you about this in this lecture. Anyway, other part of the innate immunity molecules, very, very important to get rid of the foreigners or damaged cells. We call these proteins as complements, and these complements protein, there are a lot of complements, and when they are activated, they're activated as a cascade of activation. So we call this complement system. Anyway, you only need to know complement number 3 and complement number 5 to get rid of the injure cells or infected organisms. And how it is done. On the left-hand side, on the left column, the complements can be, the complements you just recognize as only one unit that complements a lot of complement proteins. These complement proteins are stimulated in 3 ways. One is called the classical pathway. When microorganism attach with the antibody, antibody, then activate complements. This is classical pathway. There are certain circumstances that special part of the microbe, like flagella of the bacteria, directly stimulate without using the antibody, directly stimulate the complements. We call the alternative pathway of the complement. If that bacteria has special sugar like mannose, the mannose will bind with our body produce lectin. The lectin will bind the mannose of the microorganism and then stimulate complement. We call it lectin pathway. What complement can do? The complement proteins do 3 things. One, stimulate inflammation. Get the inflammation worse, get more cytokine, send signal to neutrophils. Neutrophil comes and eat, and hey, this is infection, come. That is the first function of the complements. The second function of the complements, they help phagocytes, either neutrophils or macrophage, eat the invaders or eat the injured cells more easily by using this complement and antibody coating the microbes, making like เหมือนเคลือบน้ำตาลชุบให้มันอร่อยขึ้น make the microorganism more delicious. The process of the complement antibody making the microorganism more delicious to eat, we call this process opsonization. Make it sweeter. Inflammation is like shouting, there is a food, come and eat. It's a party, for example. The third function, the third action of the complement, it forms with the membrane of the infected organism and make pores, could rule, so the cells lyse more easily. This protein complex, we call it, with the complement, we call it membrane attack complex. The first is shouting inflammation. The first is making more delicious. That is opsonization. The third is lysis the cell, making pore by membrane attack complex. These are the action of the complements in the innate immune system. Okay, I think talk everything about it. There are some other proteins that recognize microbes. Just go and read by yourself. I don't want to emphasize too much, like collectins, which recognize the collagen C1q of the microbes. Mannose binding lectins. One of them is the lectin alternate pathway in the lectin pathway in the complement. The ficolins. This has similar structure doing in the innate immune response. The pulmonary surfactant is the surfactants coating in the lung. Proteins also help promote the inflammation in the innate immunity. Natural antibodies I already described, present before infection against common patterns, and most are IgM, and they are not much specific. And those are essential functions, especially for the bacteria. The virus, when they get infected, it is more involved in the intracellular process, because the virus replicate themselves by using the nucleic acid, either RNA or either DNA. We have to have the mechanism to get rid of this foreign nucleic acid inside the cell. If we cannot get rid of the foreign nucleic acid inside the cell from the virus, we need to kill that cell, that infected cell, right away. So the innate immune, because the virus occur with us throughout the generation, throughout our evolution. So innate immunity also has some roles to get rid of the virus. The virus doesn't always wait for adaptive immunity. When we get the infection, innate immunity also has some important roles in fighting the virus, but not as good, not very good, but we have some, the innate immunity has some role fighting virus. So that's why I assign all of you 2 papers to read about the innate immunity. Let's go back to the assignment first. I haven't read these 2 papers, but I think they are very interesting. To let you be able to review that the innate immunity has some role in fighting virus. In these 2 papers, I specifically choose for you COVID-19, innate immunity. And COVID-19, through the evolution, it is interesting enough, it can evade or escape the innate immunity. So when you present, you must present these 2 points. One is how innate immunity responds to the COVID-19, and how COVID-19 evade or escape the innate immunity. That's why we need the vaccine or better adaptive immunity to fight against COVID-19. So on Thursday after like 11 maybe, or if not, maybe next week, I find some like an hour for you to present it to me, those 2 papers. You can do as a group. It's not like one by one. 5 of you present 2. You can divide that 2 for 1 paper and another 3 for 1 paper. It's up to you. Just present that. I can learn something from you. It means that you understand about the innate immunity in application. That's the lab. Anyway, just go back to the lecture. I would like to emphasize that the innate immunity has some function on the antivirus. We have the toll-like receptor, as I told you. If it acts inside and the toll-like receptor acts through the interferon responsive factor, that will cause the antiviral defense. And the antiviral defense means it produces a lot of interleukins against infected cells. Not just the interleukin, I mean the cytokines. The cytokines produced by after viral infection is not very specific. It can act locally and it can act systemically throughout our body. These cytokines for the virus actually is for many response, but especially for the virus, they are important. I think you must remember these pro-inflammatory cytokines in the innate immune response. These are tumor necrotic factor, necrosis factors, interleukin 1 and interleukin 6. And I'll tell you a little bit more. What do they do? Actually, tumor necrosis factors and the interleukin 1 has similar effects, but it's produced slightly. It's different molecules, but they are both cytokines. Similar effects, but different sources. Like TNF, it is mostly produced from macrophage and T lymphocytes. And the interleukin 1 is produced mostly from the local tissue like macrophage or endothelial cells, interleukin 1. And the effect is similar. TNF and interleukin 1, locally it activates endothelium and draw the neutrophils into the infected sites. And locally it acts on the endothelium and the neutrophil, but systemically it goes to the hypothalamus and cause the fever. The higher temperature, the infected viral cells is more unstable. So the fever is good somehow in terms of get rid of virus, but actually it's not good for our body either. So we don't like fever. Virus don't like fever. We don't like fever too. And it also stimulates several acute phase protein. It means that a lot of proteins that is produced in stress or in the infection. And it stimulates the catabolism as well. Our body like hopefully more active, but how effective it is, not many. As for the interleukin 6, they also stimulate these acute phase proteins, but apart from that, it also stimulates the B cell proliferation and therefore it stimulates antibody production. Function of these interleukins are similar. So not much. This one, I just want to let you know that these TNF, interleukin 1 and interleukin 6 has the local effects on the endothelium cells. So to increase permeability, and it also acts as a chemokine to draw the white blood cells into the injury site or infected sites. That's the local effect. They have the systemic effects, like going to the brain and cause the fever, going to the liver and stimulate the acute phase proteins, going to the bone marrow to make the bone marrow produce more white blood cells and more release white blood cells to the blood. But for the systemic effects of these pro-inflammatory cytokine, it can cause the bad effect as well. Bad to the heart, reduce the contractility, bad to the blood vessel, that cause the coagulopathy, more the clots inside the blood. And if increase the permeability too much, the fluid will leak out, you will get the swelling, and it will cause the muscle to be more insulin resistant, for example. So not always that these pro-inflammatory cytokines are good, but you need to know anyway that these are important 3 cytokines in the innate immune response. What I want to talk about is not much, maybe perhaps. Just to summarize that from the patterns of the microbes stimulate the pattern recognition receptor, and this receptor activates has two effects. One is to stimulate inflammation by acting to secretion of the cytokines. And also they have some, if it is phagocytes, it has the downstream effects on the phagocytic enzymes or oxidative enzymes to get rid of the microbes. For the virals, the antiviral defense, the inflammatory response, as I said, that the other leg of the toll-like receptor, for example, it's not just the stimulate NF-kappa-B and cause the inflammation. We already talked about inflammation now. And now on the other leg, that is stimulate the antiviral defense. It needs the stimulation via transcription IRF transcription factor, and the IRF transcription factor will cause the production of the interferon. This is another cytokine, important for the virus and important for the cancer. In this case, for the virus, interferon 1, it is very good at inhibiting viral replication. And by stimulate the interferon receptor, it activates downstream, a lot of downstream transcription of several genes. Just the term that you need to know that if the adequate stimulation of the interferons, our body or those cells, when the cells are stimulated, if you get alert not to let the DNA or outside particle get into the cell easily. So if these cells are alert and not letting the virus get in easily, we call that an antiviral state. I think so. Pretty much you know that innate immunity, most of them during the inflammation, most of the other leg is the antivirus, and the third part of the innate immunity is to send the signal to the adaptive immunity. How can the innate immunity adaptive immunity? That is by the antigen presenting cells. I already talked about the antigen presenting cell now. But then the most important one is the dendritic cell. And broadly in terms of diagram, how innate immunity can amplify signals. Send these signals and amplify the importance that we got the infection. Adaptive immunity must do something. In terms of diagrams, there are two signals that we need to amplify. How do we say? Apart from, the inflammation and the antivirus, these are the initial defense, but the innate immunity must somehow to stimulate adaptive immunity. The diagram is shown by when the antigen gets in, the antigens come from different sources. And once we recognize, we will make it more specific in later. Then the innate immunity cells will have special receptor to signal the adaptive immunity to make it learn that it is harmful and you must amplify, send more supports to us. We need the two signal. So this is the two signal hypothesis. And the signal two is important. This is the model. It's not the real. There are some examples of the real application, but this is the model. The signal to amplify the adaptive immunity, it needs the degree of the infection. If the infection comes a lot, the innate immunity will be able to send the signal to the adaptive immunity that you need to get the better and more response to it. So it responds to magnitude, and it depends on the nature of the adaptive response, whether it is how effective it is. So with this hypothesis, we can challenge and make the vaccine. It means that if we want to let the innate immunity to respond and tell the adaptive immunity to get the high level of response, we don't just add antigen into our body. We need to add some co-stimulator, or we need to add some special protein and mix together to produce the more stronger signal for the adaptive immunity. And this is still in the application. In the real life, that is the example for the vaccine. But in the real life, the co-stimulator, there are a lot of cytokines represented that hypothesis, represented that signal too. And this is the application of the vaccine. The signal to in making vaccine, protein is added and stimulate more powerful response. We call it adjuvants. Adjuvants. The terms adjuvants, strict definition means the substances added with the antigens to elicit maximal adaptive immune response. In this case, the T cell response. We can add the killed microbacteria or lipopolysaccharide or aluminum, just mostly in vaccine. Okay, about to stop in a few slides. The inflammation and antiviral defense cannot go on and on. Otherwise, we will consume a lot of energy just to defend. We cannot use our body energy to enjoy ourselves in other activity, right? So if the inflammation cannot go on and on forever, some lymphocytes and some our body can have to stop to regulate the immune response. So there are several mechanisms to regulate this, and we also need the stop signal to limit potential damage to our normal tissue as well. And this is usually achieved by the function of the interleukin. The most important one for stopping or slowing down the signal, inflammatory signal, is interleukin 10, which is usually produced by the T lymphocytes. Other cells can as well, but once you need to know is the T lymphocytes called T regulatory T lymphocyte, T reg. and also it is also produced by the macrophage and dendritic cell as well. I forgot about the Treg. Anyway, for the innate immune cells, they are inhibited by it is produced by the macrophage and dendritic cells themselves นะตัวเองกระตุ้น inflammation นะ themselves they stimulate inflammation but they know when to stop and if they want to stop they produce interleukin 10 and some other regulation of stopping inflammation may be through other cytokine secretion or the product after autophagy of their genes, their DNA parts, and they know, oh, we need to stop. We are doing too much. There are also a lot of stopping signals, but when you read the paper, there are also other proteins. These are other negative signaling pathway products called suppressors of cytokine signaling or SOCs. This is another example of signals to limit innate immune system. Yeah. And it also, once it is produced, it block or slowing down the pattern recognition receptor and reduce the production of the inflammatory cytokines. A little bit, you need to know about the SOCs or suppressors of cytokine signaling. You need to know that it is using, if you are doing a lot of intracellular signaling, the SOCs are regulated by the JAK-STAT signaling pathway. This is the intracellular signaling pathway to stop inflammation. Okay, I think I talked about the stop signals already, and these are the overview of the innate immunity. Okay, I think I better stop here. Maybe too fast, I'm not sure. So far, can you follow me about the innate immunity, or you have any question or anything I talk too fast you want me to clarify or repeat it?
Ajahn, I want to know a little bit more clear about the toll-like receptor, Ajahn. Toll-like receptor.
You need to know that let me go back to the beginning, that if you have you know, when you go to the restaurant, you have the a la carte, right? You just order and then get you what you want specifically. But some restaurants don't have a la carte menu. Some restaurants know that, oh, children must like ice cream. Oh, adults must like rice. The elderly must like noodles, for example. They are already custom made to the evolution. Our ancestor fight a long time with bacteria, with the virus. Adaptive immune response must learn that what we need, this is very special virus, we need to respond this way. We need to make this very specific antibody. But the innate immune response is act like the set menu. If this is man nose, we must have special receptor to recognize man nose, because we don't have man nose. Only fungus has man nose. If this bacteria has Flagella, oh, this is foreign. Any bacteria that have Flagella is dangerous to us. We must have special receptor to recognize this. Whether it is flagella A, flagella B, flagella C, the patterns are the same. They are flagella. We have special receptor to act against flagella. We don't care whether it is flagella 1, flagella 2, flagella 3. So these receptors are recognize this type of antigen as pattern. So we call this pattern recognition receptor. เข้าใจไหม โอเคนะ so we don't a la carte or tailor made to respond to every single. We recognize the pattern when this pattern come. It is foreign and it is dangerous to us. So this receptor we already produce since we are born because we are human through the generations. And these are pattern recognition receptor. There are a lot of pattern recognition receptor. But in this lecture, I want you to know is the only toll-like receptor. Only toll-like receptor. And the toll-like receptor is the classic one. There are like 20 plus toll-like receptor by now, I think. I don't know. What toll-like receptor do is they act they're doing as the pattern recognition receptor, as I told you, and on the other hand, they act like whether this is foreign to us or dangerous or not, or for us or not, to let it pass. So it's like the tollway or the expressway, whether you should let the normal things come in and go back behind our cell, or whether we should recognize it and destroy it. So if it recognize this pattern, this is the function of the toll-like receptor. If it recognize as it is foreign, the toll-like receptor will do two things. Toll-like receptor will do either, if you think it is bacteria, fungus, extracellular, anything extracellular, it will cause inflammation by making more cytokine, TNF, interleukin 1, interleukin 6. And if toll-like receptor think it is virus, the toll-like receptor will tell the cell that you should get rid of this foreigner as a virus. You should kill it by NK cell, or you should send the signal to making more cytokine, or sending it to the T cell, assigned by the dose of interferon. Okay. So at this point, do you understand?
Yes, John. I more understand about it. Thank you, John.
Okay. If no other question, I think of you made that ไม่รู้เรื่องเลยอะไรเงี้ยหรือเปล่า Okay. So for the assignments, have you take a look briefly on it yet?
I have read one, but not finished yet. I have read one paper among two papers.
Okay. Anyway, if you think it is too difficult, you feel free to discuss with me, no? Otherwise, I will find. I think maybe not this Thursday. I might be too busy, but maybe next week I find.
Okay. Yes.
An hour.
Okay.
Just to present briefly.
Okay.
Like 15 minutes. No more than 20 minutes per paper.
Okay.
Okay, I think I finished the class today now. So let's see you again later in class okay สวัสดี
สวัส
ส
ดี
วัส
ครับ
ดีค่ะ
สวัสดีครับอาจารย์